so the example basically used geometry to find, I'm going to call them just the initial reactor 1 and the larger one 2. Okay, so I think everyone was pretty clear on how to get the geometry numbers. So those were the geometry numbers that you use based on the 2-to-1 relationships and the volumes that you were given. The big thing that you had to realize is that you were not using the amount of cells, you were using the amount of product to most easily do your calculations. So it told you that you had 2 grams per liter of product and in a 2-liter reactor, and 50% was from each of the ones in volume and the ones on the surface. So in other words, in a 2-liter reactor, you're getting 2 grams from the surface area attached cells and 2 grams from the, I should say the surface area attached cells, and 2 grams of product from the cells that were in the bulk of the fluid, right? So, in other words, I'll just put that specifically 2 grams for 738 centimeters squared. And so to scale that up for the large S2, so that's 2 grams for S1. So for S2, you've got now you're getting 928 grams from the scaled up reactor instead of 2 grams. Okay, does that make sense to everybody? So what we're doing is saying 2 grams were surface associated, or from surface associated cells, and we're scaling up the surface by 342,000 to 600 to over 768. So the result is you make nearly a kilogram of product on the surface of the scaled up bioreactor. Does that make sense? Yeah? Okay, and then the stuff in the volume is actually much easier. So now you've got 2 grams times 20,000 liters over 2 liters. Just gives you basically 20,000 grams just from the volume, okay? So total product is 928 grams. Well, actually, I'll just write the full answer. 20,928 grams. take-home message is that in the 2-liter reactor, half of your product is coming from surface-associated cells. In this case now, like 10% of your product is coming from surface-associated cells. Right? Makes sense? Because what it was saying essentially is that in this particular case, your surface-associated cells were way more productive than your cells that were growing in suspension. So if you're scaling up your reactor, you're knocking down your surface to volume ratio a lot, and suddenly what looked like a very productive reactor becomes a significantly, like almost half as productive as the scaled up, or the small scale version. So if you just scaled it up without anticipating that there was some productivity from the surface associated cells, you would be out by a factor of 2 almost. So it is important to check it out, and that would suggest if you have such good growth from surface-associated cells, it would suggest that probably you should be exploring some kind of immobilized cell system if you want really high productivity. Okay, did that make sense? I think most of the people were most of the way there by the end of last class, but I promised I would take it up. Okay, so we're next going to do another example. Our last example for scale up, and that's example 10.3. Okay, so we're scaling up a fermenter from 10 liters to 10,000 liters. Okay, and height to diameter ratio is 3:1. Okay. Impeller diameter was 30% of tank diameter. Okay. Agitator speed is 500 RPM. And there are 3 Russian impellers. So it says determine the dimensions of the large fermenter and the agitator speed. So the dimensions of the large fermenter means what's the diameter of the larger fermenter. And then also what the agitator speed would be given different scale up rules. So the first we're going to look at is constant power to volume ratio. Second, constant impeller tip speed, so shear on the cells. And the third is constant Reynolds number. Okay. So we're going to assume that we're keeping them as both as cylinders, both with the same height to diameter ratio, but we're going to scale it up from 10 liters to 10,000 liters. So I'm going to have you skip the how to do the scale up, and have you notice that volume is cubes, and this is just 10 liters to 10,000 liters. It's just times 10 times 10 times 10, right? So that means that each of your sizes goes up by a scale of 10. I mean, it would be easy if it was a cube, but it works the same way like a cylinder. So I will get you started on the numbers because it just is tedious to calculate the numbers, and have you do the rest of the calculations. So we've got DT1, 16.2 centimeters, and so DT2 is 1.62 meters. Okay, and DI is going to be 30% of that. Okay, now, does everyone still have their notes from this morning where I said what the constant things are that you scale up for P over V, impeller tip speed, and Reynolds Okay, so then you should be able to go ahead and practice that example. We can turn off the video for the time that we're doing that. Is you end up with much faster spinning if you keep your power to volume ratio constant. It's still slower than the scaled down reactor because you've got a much bigger impeller as well. And generally, you would, if you can, you would choose a constant power to volume ratio unless you have really shear sensitive cells. So generally, you want to run your reactor a little bit below the amount where you'll get shear problems with your cells. But if you want to, like, you don't want to hit that point where you're going to start killing your cells with the tips of the impellers because of the shear sensitivity of your cells. But you want to be as fast as you can without causing problems to your cells, because that maximizes your oxygen transfer, which is really important to keep your cells alive. So it's a balance between oxygen transfer and shear problems with your cells. Truthfully, so we've just sort of went, almost all of the scale-up work we've been talking about is really only applicable to cells with cell walls. So like E. coli, yeast, and so on, bacteria. And we've had a couple of little questions about what you do with mammalian cells, and Carl's idea of geometric similarity, where you have a bunch of the same kind of reactor, actually is remarkably true with mammalian cells. You almost have to do that because they're so shear sensitive that you can't, you can do a little bit of agitation. So you'll see mammalian cell bioreactors that are sort of these small glass cylinders with a little blunt stirrer in it, and they're about this high. And you can get away with some stirring in suspension in a glass stirrer like that, basically on a little stir plate. So we're talking less than a liter of reactor volume. You could maybe scale it up a little bit more than that, but not much more than that, because then you start having shear sensitivity problems with your non-protected, no cell wall mammalian cells. So actually, the way they grow mammalian cells in pharmaceutical companies is they have these plastic bags on rocker plates, and in suspension, when they're trying to grow it in suspension. And they basically fill the plastic bag with cells and medium, and then they rely on diffusion through the plastic bag from the surface to give you enough oxygen transfer, and it just rocks the cells back and forth to keep them in suspension and to keep them near the surface of the bag, so that they are at the surface of the liquid, so that they get enough oxygen transfer to keep them alive. So really low tech, like not very well scaled. They just have lots and lots and lots of these rocker platforms and incubators. Not really well scaled. So as a chemical engineer, if you could think of a really good way of scaling up oxygen transfer and without causing lots of shear stress to cells, you could probably cut down the price of antibodies significantly, because most antibodies can only be grown in mammalian cells. You could cut down the price of some really big recombinant human proteins that can only be grown in mammalian cells. And they're really expensive, really expensive. We're talking 0.1 grams for $400. So, and that's because it's so difficult to scale them up. So if you could think of a good way of doing it that's better than lots and lots and lots of plastic bags in a big incubator, you could probably make a whole lot of money. So get your brain hats on and start thinking. But the problem is that bubbles don't suit mammalian cells. Lots of agitation doesn't suit mammalian cells. You actually have the same problem trying to think about how to grow up lots of stem cells for regenerative medicine, because say you want to make a regenerative, a tissue engineered liver, that's billions of cells that you need. Cells are packed really tightly. You know, even if you extract like that much cells to make that much liver, you still have probably 100 million cells minimum. A lot of cells. So scaling stem cell production up so that you can use them for therapeutic purposes is actually a really big area of research as well. Lots of people are trying to use various immobilization techniques so that they can basically proliferate within small beads, and the beads keep them from being experiencing shear stress. So they're sort of grown within a little matrix that is sort of like an extracellular matrix, and then the outside of it is the part that experiences shear, but within it, it doesn't have much. So you can treat the bead a lot more harshly than you can cells. But it's a tough problem, scaling or having a million cells and scaling them up. Any questions about scale up? Okay, also I have posted assignment 4. It is due the 14th of December, so you've got lots of time to do it. If you've had a chance to look at it, the last question on assignment 4 is actually not due the 14th of December, it's due the 10th of December, but it's a bonus question. So if you want to get 1% free on your course, you develop an exam question and complete a solution for it. And post it on the Avenue discussion board.
Really easy.
If it's, so I will, it's not an automatic 1% just for doing it. If it's crazy easy and kind of ridiculous, then you'll get the 0.5% for doing something that's still relevant. If it's crazy hard, then I'll give you congratulations, but definitely won't use it as an inspiration for an exam question. None of them will be used directly as an exam question, but I might use some as, if they're good ones, I might use them as inspirations for exam questions. But it'll also hopefully give people more practice problems to try, is the real incentive. So I actually find if you, the exercise of going through trying to create a problem can teach you as much about the problem as anything else. Just like teaching it teaches you the material better than learning, like sitting there absorbing it. Teaches you the material. So there you go, there's one question that's due earlier, but it's just for bonus. So you don't have to do it, but give it a try. And I picked December 10th because that's 2 days before my exam copy is due. So if someone has a particularly good one, then I can use it for inspiration. No, I won't have any carbon copies on the final exam. I promise you that. Okay, any questions about that? Okay, so I think I'm gonna talk a little bit about instrumentation for bioreactors. And I think I'm gonna end the class just a teeny bit early for you because I know you have an exam. And then on Wednesday, we'll talk about sterilization. Okay, so if you're instrumenting a bioreactor, putting, like we talked about this in measurements, putting instruments on a bioreactor, a lot of you also have done the fermenter experiment in 4L2. What are the minimum sensors you would want on it, and what other ones might you think about? So what would you need to monitor in a bioreactor and potentially control?
Temperature?
Temperature is obviously critical. We have learned that your growth curve is really dependent on what temperature it's at, and that cells make a lot of their own heat. So if you're not measuring the temperature and controlling the temperature, who knows what weird stuff's going to happen to your culture. So temperature is critically important. And usually it's one like a thermistor and RTD type sensor that's on there. Okay, what else? pH is obviously also really important because the cells will, on their own, will dramatically change their own pH if you don't have some mechanism to change the pH back to neutral. So you need a control system that measures the pH just with the pH probe and then has the facility to bring it back to neutral. So temperature and pH are really important ones. What else?
O2?
Yeah, dissolved oxygen. And as you know, dissolved oxygen sensors are on the finicky side. Don't always give you the best results, but it's really, again, where you see it being particularly important is when you get to the high culture volumes, or high culture concentrations, and the cells are rapidly using oxygen, and you start getting in an oxygen-limited situation. So you may or may not be controlling for dissolved oxygen, but so actually one of the things, so in 4L2, you're tasked with changing reactor conditions to affect how your cells grow and how your product grows. And especially the biology students tend to really just want to, they have the things that they can adjust, and so they just want to adjust the things that they can adjust. So they like saying, okay, well, we have a sensor for temperature and we have a controller for temperature, let's change temperature. Well, for the most part, at least with growth, that's a pretty predictable response. Some products are sensitive to it, but most cells, it's very well known how cells respond to it. They'll also want to control for pH. Again, for the most part, how cells, E. coli responds to pH is very well documented. So you're not necessarily learning anything new, and if you're departing from a very well established optimum pH from the literature, the only way you're going to go is worse. So you're trying to get better. So if you're departing from best, the odds are that you're not going to get better, except in those cases where it makes your product form more accurately. You can slow down the production by doing things like decreasing temperature. You'll also see students who haven't taken a bioreactors course saying, let's just have 2 different impeller speeds or 2 different sparge rates, and we'll keep that constant. But what the cells see isn't constant if you do that in a batch reaction. So if you're keeping pH constant, what the cells see is constant. If you keep temperature constant, what the cells see is constant. But in a batch type reaction, or even to some extent in a fed batch reaction, if you keep your impeller speed or your sparge rate constant, what happens is the dissolved oxygen level starts at saturated and stays at saturated for a long time until the cells get really dense, and then all of a sudden your dissolved oxygen plummets because they get too dense and they're using up all the oxygen more quickly than it can come in, and they become oxygen limited. So really, as far as the control mechanism from the cell's perspective, in a batch reaction, you're not keeping anything constant by keeping your sparge rate constant or your impeller rate constant. So I would encourage you not to consider doing that. And maybe you can do a manual control on dissolved oxygen. You always want your dissolved oxygen to be above something, above 0. So at the end of a batch reaction, you may want to increase your sparge rate or your agitation rate. What they become concerned about is that you're above, you may start shearing the cells, which is a valid concern. If you're really upping your agitation rate to the maximum, you now have means that you can figure out what the rate of you can go and measure the impeller, the diameter of the impeller, and you can figure out what the fastest thing is, and you can figure out about what your tip speed is going to be for your impeller, and figure out the shear on the cells. So you can do a quick lookup of how much shear E. coli can stand, and are you even getting near it by going to the maximum agitation rate? Probably not. E. coli are pretty hardy things. So it's possible that you start damaging them if you go to the very highest agitation rate, and that's a valid concern. But again, if you're going to a really high agitation rate, all you're doing is getting out of that optimal growth area, and you're not going to increase production. You just might get higher oxygen. Like if you keep it high through the whole thing, there's no point doing that because you've got lots of oxygen for the start of the culture. It's just near the end you have to start worrying about it. Anything else that you might want to monitor? So those are the simple ones that you sort of need to monitor, just to understand what's going on. The time is, especially if it's not a CSTR type reaction, is very important. Yeah.
The airflow.
So that's sparge rate. Yeah. Yeah. That goes right along with the agitation rate. So that it's so you definitely have flow sensors that tell you what your sparge rate is, and that's important because you may turn it up or turn it down. And you have a sensor that tells you how fast you're spinning your agitator, because you don't want to just go and say, I don't know, it's maybe 700 RPM, maybe 200. High, it's high. And if you're doing fed batch, you want some sort of, you'd ideally have some sort of flow sensor. If you do a fed batch in 4L or 4LL, you actually end up, there's no flow sensor, so you end up having to calibrate a peristaltic pump, which means, anyone know what a peristaltic pump is? Yeah, lots of nods, okay. So it's got, did you use it in 4L? Yeah, so it's got the tube, so it depends on what your tube size is that's going through it. So you have to pick what tube size you're going to use, and then stand there with a stopwatch and a cylinder and figure out how long it takes to fill up 100 mils, and then you get your flow rate by time. So we're not super high tech in 4LL. And you'll probably actually find that the flow rate you want can't be quite, the peristaltic pumps that we have can't go quite that low. So instead of having a nice smooth continuous flow in like we've done the modeling for in fed batch reactors, you'll probably have a bit of a stepwise pattern. But it's close, right? It approximates it. It depends how much you care to get close. You could be really anal and do it, for 1 minute on, 1 minute off, 1 minute on, 1 minute off. Most people do it for, 2 minutes on and then 14 minutes off or something like that. I can't speak for the particular pumps and flow rates you're going to use, but okay. So flow is obviously another one that's important. Gas flow, and if you're having any liquid added liquid flow. What other parameters? Yes.
Cell concentration or product concentration?
Yeah, so cell and product concentration are convenient if you can measure online. Typically it's not, it can be difficult to measure online. In the lab course, you can't measure it online, so you take samples out every 15 minutes and just do the simple OD measurement that I think you did in 4L as well. So it's not perfect online. I think you can get sensors so that you can do online OD measurements. Bubbles can interfere with it, and you have lots of bubbles. So I think it's not always done, but it can be a helpful thing. Yeah.
Can you measure emissions, like CO2 or?
Yeah, so you can put sensors on the gas emissions, and that can actually give you a lot of information about, and that's what we were talking about when you measure the KLA. You may want to do that. But beyond doing that, it can give you information about actually the number of cells. That can be an indirect measure of the number of cells, because the oxygen consumption rate is related to the cell number. So if you can calculate how much oxygen is consumed, it can give you information about what your cell number or cell concentration is. So some people will do that as an indirect method of doing online cell concentrations. Anything else you can think of? I think that's a lot of them. The other one actually, so you can, I said you could do online turbidity measurements for cell concentration. Sometimes people will check the viscosity, but rarely online. That tends to change as the cell is making lots of product, depending on what kind of filter you have. But the other one that really gets measured is foaming, because that can really interfere with your process. If you have a lot of foam suddenly building up, then suddenly all your filters get wet, and they don't work anymore. So that's problematic. So you want some sort of sensor that tells you it's time to break the foam or add more anti-foam. In the lab scale reactors, you peek in and look to see, which is why glass is so convenient. You can look and see if there's foam in your eyes or your sensor in that case. But you can have in much larger metal vats, you can have online sensors. Typically, the output of those sensors is very difficult. I mean, you can do direct control of temperature and pH, and even oxygen, but it can be difficult to do direct control of things like product concentration. So it tends to be more of a make sure that the process is working properly situation. Okay, so next class we'll talk about sterilization. 6 minutes early. Any last questions? Good luck in your exam.
Feels
Wednesday.
good.